Okay, our second video here, and it's going to be on the reception. So if I quickly show you your outline, we've already gone over Roman numeral 1. Okay, that was just our inner, talking about signals, how they can be locally or long distance, and then talking about the 3 stages which are represented here: reception, transduction, and response. And now we're going to go into detail about those receptors. Then we'll go into detail about transduction and finally about the responses. Okay, so when it comes to reception, when those signals come along, there's going to be one of 2 things that's going to happen. It's either going to attach to one of those catchers on the membrane, or it's going to go right through the membrane. Let's start with the ones that go right through. The ones that go right through are going to have to enter into the cytoplasm. They enter into the cytoplasm here. Okay, because we are on the outside of the cell here. It's going to have to travel across. And what's the only thing that's going to get across that phospholipid layer? Just things that are small molecules and non-polar. Okay, like I like to say, like freshmen, they got to be small and greasy. Remember, non-polar is the opposite of polar, so that would be more like an oily substance. And then the receptor is going to be deep within the cytoplasm, okay? And that's pretty much all about that. Remember, though, with all these receptions, this is like a key and a lock type of thing. So if the key is small and oily, it's going to go through, but then it's got to fit into that lock. That catcher and that signal are going to be very specific fit, like a key in a lock, like a puzzle piece. Okay, and so that's your intracellular thing, happens within the cell. But most of your catchers are going to be on the cell membranes, and they're going to be the proteins in the cell membranes. Now, I drew these proteins all differently because that's kind of how they are. All these proteins are shaped differently, but the 3 that we're going to talk about, and they're really, I don't know of any others, but these are the 3 I know about: G proteins, tyrosine kinases, and ion channels. These are all catchers, if you so to speak. They're receptors, and they're all proteins that are in the cell membrane. So we're going to start with the G proteins. These are actually the most common. Okay, and they're a crazy looking molecule. Of course, this is not exactly how it looks like, but they usually have some sort of part where the signal, okay, the active site, okay, the signal is going to attach into. It's like kind of like the lock, and the signal is the key. Then it has this really like crazy structure to them, but then it has like another section outside that. And we'll get into more detail about how the G proteins work. Okay, tyrosine kinases, this is about as much detail as we're going to get in that I say here now, are 2 molecules that have like 3 little nubbies on them a piece, and they also have the active site part sticking outside. When a signal, and you got to have 2 of them, when 2 signals come along, these 2 molecules will kind of merge together, and ATP will start to phosphorylate them, which means on each little nubby, which means you'll get 6 responses. 6 responses. So I like to call the tyrosine kinases the multitaskers. Okay, they're going to get a lot done. Not that these others aren't going to get a lot done, but usually we have 1 G protein associated with 1 transduction or pathway. And with tyrosine kinases, we'll see 6 pathways coming off of that. But they could only do 2. They could only do 1 at a time. Okay, but they have the possibility to do 6. And finally, the ion channels. When a signal comes along, and the signal, I should get my black pen here, the signal, just like the others, comes along, it will like kind of attach, okay, the signal comes along and attaches to the protein, and it causes this gate to open up. When the gate opens up, the floodgates open up, the ions start to flow through. Okay, you're going to see that especially in the sodium-potassium pump, which is in nerves and muscles. But they're very common. You also have heard of these ion pumps when we talked about photosynthesis and respiration. Okay, those hydrogen ions that were being pumped into the thylakoid space for the light reaction, or the hydrogen ions that were being pumped into that inner membrane space of the mitochondria and then diffused through to make the ATP. So they also may have had signals that come along. They definitely did have signals that come along that opened up those gates. Okay. So there are four types of receptors. Three of them are in the membrane. One's then deep within the cytoplasm. And that's it for reception. But we are going to go into detail in the next video on G proteins. Okay.